This is the second tutorial on how to use TurboSM software. We will focus on some basics on how to view and analyze data. After recording, the data file will be automatically read in. You can also read other previously recorded data files here. File, Open File. You select file you want to view. The data file contains a series of images. The image displayed in the top window is annotated here, frame 51 minus dark frame, in this case. As the cursor moves around inside the image, the value and the coordinates for each pixel are displayed here. Now let's select a region of interest by dragging a box anywhere on the image. The waveform from this region of interest is displayed in the bottom window. Well, there seems to be bleaching effect in the fluorescence signal. Let me reduce the Y gain, the vertical magnification, so we can see the whole trace. There are many ways of selecting a region of interest. Dragging a box is default and a quick way to do it. But in most cases, it's better to select the drawing pen here, and then trace around the cells or any structure you want to trace. Right-click to clear all regions of interest. Then let's trace around this cell. Drawing pen allows you to draw irregular shapes. Again, here is the waveform from the region of interest. We can see many downward spikes on this trace. The polarity of the spikes usually depend on the type of voltage probe you are using. We can invert the polarity of these action potential spikes here to make it easier to view and analyze. Let's first add some high-pass filter to get rid of the DC drift. Then we can increase the gain a little to see the spikes. Finally, a bit of low-pass filter to get rid of the high-frequency noise. Now, you can see many action potential spikes with decent signal-to-noise ratio. Let's add another cell. Then adjust the gain to your liking. Okay, now, let's take a closer look at these buttons on this main interface. This section is for movie control. Previous frame to go back one frame. Play to play the movie of this time series. Pause to pause the movie. Next frame to go one frame forward. This button inverts the polarity of the waveforms. This section has tool for selecting regions of interest. Click Kernel allows you to select, by single click, circular regions of interest with diameter in pixels defined by the red number inside this circle. You can change the diameter with these two arrows. Drag a box to draw a rectangular region of interest. This mode is currently selected, and therefore the text is in red when the cursor is hovering over it. The button of selected mode is always highlighted. Draw area to draw irregular shapes. Remove to remove part of existing region of interest. Add toroid is a checkbox for add more pixels to the current region of interest. It's currently inactive because the box is not checked. Threshold is another checkbox. When it's checked, you can click on pixels of certain brightness and areas of pixels with the similar or greater brightness will be selected as a region of interest. When the cursor hovers over the slider of image scale, you can see the max and min values used for scaling the current image. When auto scale is checked on the right panel, the max and the min scaling values will be automatically calculated for the image displayed. By clicking on the slider or the two pairs of arrows on both ends, the auto-scaling will be switched off and you will be setting the scaling range manually. To display different frames, you can click or drag anywhere along the traces here. The frame number below the image should always correspond to the frame position marked by this white line. The numbers next to the white line are frame number in white and data value versus dark or reference value in corresponding colors of the traces. The values in the histogram are also color-coded and correspond to the colors of regions of interest on the image. The values offer a glimpse on how bright your sample is. Be aware of that the brighter samples give better signal-to-noise ratio. Normally, the values in the trace window should be the same as the histogram, if the traces aren't filtered or inverted as shown here. Finally, I would like to point out the information about the data file is displayed up here, including file name, time of acquisition, configuration and a number of frames. There is an arbitrary limit how much data to read into the memory in one read. If the data file is larger and only part of it is read, you can read the next section by clicking on this arrow. That's all for now. We will talk more about the display settings in the next session.